Hi, my name is Ryan Donnelly, and I am a product manager at Canon BioMedical. I'm excited to share a quick and simple protocol for determining the copy number of the SMN1 and SMN2 genes. This webinar will explain the scientific principles behind our method, share an overview of the workflow, and touch on how data are analyzed for our Novallele copy number assays. Before I start, I need to share a few disclaimers. First, the products discussed in this presentation are for research use only. Licensing information can be found in the product-specific user guide, which is on the Canon BioMedical website. Our Novallele assay products are available directly from Canon BioMedical in the USA. Outside of the USA, our distributor network makes our products available in select countries. Now that that is out of the way, let's get started. The Novallele copy number assays determine copy number changes of specific genes. In this example, the SMN1 and SMN2 genes. The wild type is having 2 copies of SMN1, as shown in the top rectangle. The assay can detect heterozygous deletions, which leave 1 SMN1 copy, or homozygous deletions, which result in 0 SMN1 copies. Inversely, the assay can also determine if a duplication event has happened and detect 3 or more copies of the gene of interest. From a biological perspective, both the SMN1 and SMN2 genes produce the same protein, SMN. This is due to the nucleotide sequence between the two genes being almost exactly the same. The only difference between the two genes is a single nucleotide in exon 7. This minute difference, however, alters the splicing patterns of the messenger RNA of the two genes. While they both code for SMN, the gene splicing pattern of SMN2 produces full-length functional SMN protein only around 10% of the time. The remaining 90% is truncated and is non-functional. While the homology between the SMN1 and SMN2 genes creates a backup for SMN protein production, the homology also makes designing assays for these two targets quite difficult. To ensure the robustness of design and amplification of the correct gene, we conducted extensive testing using numerous SMN1 and SMN2 copy number controls that had been previously characterized. When we set out to create copy number assays for these two genes, we wanted to simplify the protocol as compared to other methods on the market. The Novallele copy number assays use high-resolution melting, or HRM, to determine the copy number of the gene of interest. When performing the protocol, purified genomic DNA is mixed with the Novallele copy number master mix and the copy number assay. A great feature of both assays is that they have the same cycling conditions so that they can be run on the same plate. The reaction mixes are run through a standard PCR cycling protocol, and at the end of cycling, the products are melted and the data are collected. As the temperature increases, the change in fluorescence is captured as the PCR product melts. I will go into more detail of this later. Results can be analyzed using the Novallele HRM Analyzer software, which I will also touch on later. There are a number of different HRM-enabled thermocyclers available from such companies as ABI, Biorad, Roche, Qiagen, to name a few, and you likely already have one in your lab. In summary, the Novallele copy number assay protocol is accurate as proven by extensive bench testing of the chemistry. It requires minimal effort since the PCR and detection take place on a single instrument. It is quite cost effective as only common PCR reagents are used. It is simple as you don't need to run technical replicates to get quality copy number calls. And lastly, it is very fast, especially compared to other methods. Just how fast, you may ask? 2 other commercially available methods for SMN1 and SMN2 copy number determination are droplet digital PCR and multiplex ligation-dependent probe amplification, or MLPA. Novallele copy number assays generate results in around an hour once the reactions are prepared and loaded onto the thermocycler. With specific thermocyclers, for example the ABI Quant Studio, data are available in less than an hour. Droplet digital PCR, on the other hand, will take 2 to 3 hours to generate results for just a few samples. The runtime is dependent on the number of samples. Large batches will take longer than shown here. In addition, this method requires specialized digital PCR instrumentation and components. MLPA is a 2-day workflow due to the lengthy hybridization step. In addition to the long protocol, MLPA also requires that the lab have access to a capillary electrophoresis system in order to generate the data. As I touched on earlier in the presentation, the Novallele copy number assays use high-resolution melting to detect the copy number of the SMN1 and SMN2 genes. For those of you not familiar with high-resolution melting, here's the basic premise. A nonspecific intercalating dye that only fluoresces in the presence of double-stranded DNA is a fundamental component of the reaction. At the end of PCR, the fluorescence value is at its highest point, At this stage, the temperature is raised at a linear rate to denature the double-stranded DNA, and all the while the fluorescence decay is monitored by the optics inside the thermocycler. Each possible copy number yields different fluorescence signatures during the melting and are therefore able to be separated from one another during data analysis. While HRM can be used to detect SNPs, quantify changes in methylation levels, and characterize genomic repeats, let's look specifically at how HRM is used to determine copy number of a particular gene of interest. In order to use high-resolution melting for copy number variation, there are two key components related to assay design that must be understood. First, relative fluorescence values must be maintained to segregate samples with different copy numbers. The relative fluorescence is locked by not allowing the reactions to reach the plateau phase of amplification. If the reactions were to reach plateau, the fluorescence differences of the target genes would be lost. To keep the reactions in the linear amplification range, the number of PCR cycles is limited to 25. The second key component of assay design is related to amplification competition. Primers for a 2-copy conserved reference gene, as well as a primer set for the target gene, are included as part of the reaction mix. Amplification competition is created by limiting the available dNTPs in the reaction mix. This affects the duplex reaction of the target gene, in this case, the SMN1 or SMN2 gene, and the reference gene. You will notice in the graph shown on this slide, that there are two melt curve peaks. One melt peak for the target to the left, which for this example is SMN1 at the lower temperature, and one melt peak for the reference gene, which melts at a higher temperature and thus shows up on the right of the target gene melt curve. One thing to note is that the reference gene is a known 2-copy gene conserved in the genome. Knowing this, you may wonder why the reference gene peaks vary when 2 copies of the reference gene are in every sample. This is due to the competition between the duplex. The variation in reference gene peaks is related to the number of primer binding sites available for both the target and the reference genes. The black curve, which represents 0 copies of the target, has the highest reference gene fluorescence value because of all the dNTPs are available for the reference gene, since there is no target gene available to amplify. Inversely, the lowest reference gene fluorescence value, the green curves, are from samples with 3 copies of the target gene. So the target gene has more primer binding sites. The 3 SMN1 gene copies compete against the 2 copies of the reference gene. We set up the reactions as competitive because we get another way to separate samples with different target gene copy numbers. Not only can you look at the fluorescence differences of the target gene melt curves, but you can look at how the reference gene melt curves are affected by the copy number of the target gene. Using the fluorescence values of both the target and the reference genes creates a fluorescence signature that easily clusters samples. Now let's dig into how data analysis works and ultimately how copy number calls are determined. As mentioned on the last slide, the combination of target and reference gene fluorescence values create a fluorescence signature unique to the samples with the same target gene copy number. For samples with the same copy number, this signature is distinct enough that they group together. Running a panel of characterized controls with different copy numbers for the gene of interest, along with your samples, makes data analysis very simple. The Novallele HRM Analyzer software, which clusters samples with the same copy number together and visually distinguishes them by color. To make a copy number call, you simply confirm which one of the characterized controls is clustered with the sample. For example, if a sample clusters with the 1 copy SMN1 control, then that sample also has 1 copy of SMN1. To aid in the analysis, we optimize these assays to work with both controls from the Coriell Institute of Medical Research or controls that have been previously characterized in your own lab. You just need to make sure that any previously characterized samples you used were prepared using the same DNA extraction method as your test samples. One more item to note is how the data are presented. The data we showed on the previous slide used a derivative curve to show the 2 different melt peaks, but I think showing the data using the difference plot, the graph on the right of this slide, makes visualizing the different clusters easier. The data are the exact same, just transformed using a different algorithm. One of the nice things about using the difference curve is that you can select a specific cluster as the baseline, which flattens this set of data. The example on this slide has the blue one copy curve as the baseline, and all the other samples are compared to this cluster. Samples with fewer copies of the target gene show up below the baseline cluster, and samples with more copies of the target gene show up above the baseline. One of the limitations in using relative fluorescence to separate samples of varying copy number is that the fluorescence differences shrink as the copy number of the target gene increases. While you would expect the target's fluorescence of a 1-copy sample to be approximately 50% of a 2-copy sample, the difference gets much smaller when comparing a 5-copy sample to 6-copy sample of your target gene. For this reason, both the SMN1 and SMN2 Novallele copy number assays can distinguish samples of 0, 1, 2, and 3 or more copies of the target gene. Any sample with 3 copies or more of the gene can be called as such since the fluorescence difference at those higher copy numbers is difficult to differentiate. You can see in the SMN2 graph on the right how close the 3 and 5 copy controls are to one another. Now imagine also running a 4 copy control that would show up in between them. For those of you that need to differentiate higher copy numbers of these genes, a digital PCR approach is your best bet, as that technology can return such analysis. However, if you need a simple method for calling 0, 1, 2, and 3 or more copies of SMN1 or 2, then the Novallele copy number assays are a great fit for your work. The sample data shown today were visualized using the Novallele HRM Analyzer software. At Canon BioMedical, we have a lot of experience with HRM analysis software that comes loaded on HRM-compatible thermocyclers, as all of our Novallele assays use HRM analysis. We developed our own software for HRM analysis that is very intuitive and user-friendly. Melt data from many of the common HRM-enabled thermocyclers can be seamlessly uploaded into the Novallele HRM Analyzer software, and for those systems that we haven't worked with yet, our scientific support staff can help you put that data into a format that can be uploaded and read by the software. The Novallele HRM Analyzer software features data normalization functions, clustering algorithms, data exporting, as well as image capture capabilities for use in posters and publications. The Novallele HRM Analyzer software is cloud-based. You only need to fill out a short registration form to use the software. The web address shown here, software.canon-biomedical.com, or you can find it on the Canon BioMedical website. If our Novallele copy number assays seem like a fit for your research needs, it's really easy to get more information and place an order. In the US, please contact us directly by emailing us at contactus@canon-biomedical.com or call us at 844-CANON-BIO. If you're not in the US, but are in one of the countries listed on the table on the right, please contact our distributor for that region. That's all for today. I hope that this presentation was educational. If you have any questions about our Novallele copy number assays, please send us an email or give us a call. Our scientific support staff is here to help. My name is Ryan Donnelly, and thanks again for taking the time to listen. Bye for now.